Are we good to go?
Yes, sir.
Okay, good evening. So today we're going to be doing a new type of session where we'll be discussing a pattern of mock question that has been prepared by Oracle app. So we'll be discussing MCQs, short answers, long answers, as well as essay type of questions. Okay. Um, so we'll just take a look at question paper. Now it's out of 100 marks and you'll have 3 hours for it. So some of the basic things I want you to go through while before writing an exam is to understand how to spend your time writing the paper. So here you have section A with 10 marks, section B with 5 marks, section C with 3 marks, and then we have your section D with MCQs. So it's always better to start with your MCQs first. Don't dwell too much time on it though. Maximum 10 minutes, 1 minute per each question. Okay. So once you finish that off, then you have 3 mark, 5 mark, and your 10 mark questions. So one of the problems I find students always facing is that they tend to spend too much time on your 10 mark question that they do not have enough time to write properly the remaining questions because there are a lot of these questions which are available. So make sure you don't do that mistake. You have to time yourself accordingly. I would say for a 10 mark question, 10 mark question, try not to spend more than an hour. Okay, so 10 minutes you're going to spend for your MCQ, not more than an hour to 1 hour, 15 minutes for both these questions together. So that will give you roughly 30 to 35 minutes for each. Okay. And the next, 1 hour, 40 minutes, give or take, you can spend writing the 5 mark questions and your 3 mark questions. Okay. So again, if you look at the mark basis, this is 40 marks. So that's what I want you to understand. Don't spend 2 hours writing a 20 mark out of 100 mark and ignore these 40 marks and 30 marks, which are making up the major chunk of the question paper. Okay, so small things like that. And it'll be much more easier if you could, have a proper structure when you write an answer. Each answer, there should be some heading, some proper structure. Okay, don't just, fill up entire pages of just words, paragraph after paragraph. Even the person who's correcting the paper will get really frustrated if they're correcting 150 student papers and all that. Okay, so make sure to give a proper structure to it in various headings, with diagrams that will fetch you more marks with less time taken to actually write the paper. Okay, so we can start off with the MCQs first. Okay, so again, as I was telling you, first start off with the MCQ. So we have the first question, which of the following is not a component of homeostatic control mechanism? Okay, so here we have a receptor, effector, stimulus, control center. Now, this can sometimes be a bit of a confusing question for some students. Okay, the question has been made in such a way. So, when you talk about homeostatic mechanism has usually some kind of stimulus which is coming, okay, or a change, whatever. Okay, that change, once it is coming, that is detected by something. Okay, so that detection is by something like a receptor. Now this will take it to a higher control center. Okay, and that control center will take it and give it to the effector, which will make sure it brings about the change and returns it back to its state. Okay, so in our body, your homeostatic mechanism involves this receptor, the control, and the effector. The stimulus is not under our control. Okay, most likely it cannot be under our control. So any change that happens, your homeostatic mechanism with its receptor, its center, and the effector takes in and controls it. So I'm telling you this because some students, when they look at this question, they might go for the answer stimulus because stimulus and receptor, all of that might seem similar. Okay, I'm just guiding you because these are common mistakes that I see students making. All right, so here the answer would be stimulus because that is an external factor, not inside the homeostatic control mechanism. Okay, so which is not, this is the answer. Now, the plasma membrane is primarily composed of. So this is a pretty straightforward answer. It's proteins. Around 55% is your proteins and 40 to 44% is your lipids and less than 4% is your carbohydrate. Okay, so of the lot, the major is your protein content. This is actually not a very good question. Gap junctions are most commonly found in. Okay, so even as an educator, I found it a little confusing and difficult to make sense of what exactly is the question here. So the thing is, gap junctions are commonly found. It's there everywhere, except adult skeletal muscles and your erythrocytes. Okay. Apart from that, most places in our body have gap junctions. Now, most abundantly present, you can say smooth muscles are there and your cardiac muscles are there. But again, if you look at the nerve cells, the nerve, in terms of its electrical synapse, in terms of its connection with glial cells, uh, connection between the different glial cells. So, and if you just simply look at the number of cells in the entire body versus the number of neurons which is there just in the brain, which comes to billions. Okay, so that by itself shows that the gap junctions are going to be so much more higher in your CNS than elsewhere. Okay, so I'm not exactly sure because I haven't found any review articles giving proportions, counts of gap junctions. I've gone through various articles. There is no literature I can find as to how many gap junctions, and it's in different animal models, whichever is there. Okay, so how much you can apply it to human beings also, that is another thing. So again, this is not a very good question. You can either say muscle cells or nerve cells. I would personally go for the answer of nerve cells. Okay. But I have a feeling that some people who made the question most likely would have gone for something like muscle cells as in the key. Okay, again, I cannot help you. I'm just giving you all the facts, so you can later counter all of these questions. Next is apoptosis. This is again a straightforward. It is a programmed cell death. Okay. We'll talk about apoptosis because one of the question is about apoptosis. Okay. Resting membrane potential of a typical neuron. Again, not a very good question. Typical neuron, what exactly is a typical neuron is not specified here. What we understand is we have small neurons, large neurons. Generally, we say smaller neurons have resting membrane potentials in the range of minus 70 millivolt. Larger neurons have minus 90 millivolt, but this has a big range. Okay. So again, what exactly did the person who made the question think of when they mean typical neuron is not clear. But then again, I go for this answer. Okay, so as I was explaining, majority small neurons, very large neurons are less in number. So you can go for this answer. And other components like your skeletal muscle, cardiac muscle, all of that is going to be minus 90 millivolt. If the question was smooth muscle, the answer would have been much lesser, uh, where you would have had a range with a median of minus 55 millivolt. The main cation in the intracellular fluid. So inside the intracellular fluid, the positively charged ion, which is maximum, is potassium. This is also a straightforward answer. Okay. Instead, if the question was, what is a major cation in the extracellular fluid, it's going to be sodium, which is a major anion in the extracellular fluid is chloride, which is a major anion in the intracellular fluid. Protein is not the answer. Okay, we generally say in Gibbs-Donan equation, okay, Donan equilibrium, we always tend to talk about proteins being large structures which cannot pass through, hence the membrane potential is created. But phosphate is, in concentration wise, phosphate is higher than your protein content inside. Okay, so there, intracellularly, if the question was about the major anion, then the answer would be phosphate. Okay. Which of the following cells is not involved in immune response? So out of the lot, you can say erythrocyte. Erythrocyte do have some sort of immune function, helps in immune function, but compared to all of these major stalwarts in immune response, you can say that is the best option. This is again next question. Simple diffusion does not require. That is again very straightforward. If it required ATP, because it has to move against concentration gradient, it would have been active transport. Okay, simple diffusion is a type of passive transport. Which of the following body fluids has the highest protein content? For this, you'll have to think a little. Okay, we have plasma, interstitial fluid, lymph, cerebrospinal fluid. Now, what you have to understand is things like lymph, cerebrospinal fluid, all of it is an ultrafiltrate sort of your plasma. Okay, so whatever is there in the plasma in various forms gets filtered and taken to these. So in no way, under normal circumstance, can protein content be higher in lymph or your cerebrospinal fluid than plasma usually. Unless certain infections happen and all that, that is a completely different picture. Otherwise, in the normal scenario, plasma tends to have higher protein content. Now, thing is interstitial fluid. Okay, so when we talk about extracellular fluid, we have the interstitial fluid and the plasma. Plasma, which is stored in the blood vessels, lined by your endothelium and then basement membrane. Okay, so because of all of that, large particles like your proteins do not easily pass. So as a result of that, interstitial fluid and plasma generally have similar ionic composition, but protein is lesser in your interstitial fluid. So here the answer is going to be plasma. So even if you did not know the answer for this, if you thought a little properly with concepts, you would have got to the answer. An increase in the osmolarity of extracellular fluid would most likely result in. This is again simple. Increase in osmolarity of ECF. Okay, so outside, if increase in osmolarity is there, fluid will go to that area. When fluid goes to the area, cell will shrink. Okay, so with that, we finish the MCQs up. Now, my next thing would be to go for the 5-mark questions, because they are of more higher marks. Okay, so first question, describe the structure and functions of cell membrane. So I'll tell you what exactly you have to write over here. What is the structure and the functions of the cell membrane? So if you talk about the cell membrane, then in this question, you'll have to talk about first structure. Okay, under structure, you have to talk about your fluid mosaic model. Explain what is the fluid mosaic model. Draw a diagram. Okay, why is it fluid mosaic model? Because in your whole lipid structures, the zigzag patterns of as a mosaic pattern, you can see protein structures embedded inside. Okay, then you can describe the structures. You'll have to talk about your protein content. You'll have to talk about your lipid content. You'll have to talk about your carbohydrate content. Do this beneath the diagram. Okay, here you have to talk about protein. So this is a 10-mark question, mind you. That is why you'll have to go more in detail, because the question that is there is describe the structure and function of cell membrane. Okay, so not 10 marks, sorry, 5 mark. Okay, so out of this, what generally how we would split the answering of this, we'd give 3 mark for describing the structure and another 2 mark for the functions. So protein, what are the proteins? You can talk about the integral protein. You can talk about the peripheral proteins and what are the functions of them. Then you can talk about your phospholipids, draw the structure of phospholipid. Then you can talk about other structures like sphingomyelin, okay, sphingos and all of that. Okay, galactoserythrocytes. So like that, other various lipid structures, and okay, importantly, I forgot to mention cholesterol. And what are the function of each of them? And then talk about carbohydrate, which are the type of carbohydrates which are there, like proteoglycan, glycoproteins. Okay, so like that, you mention the different carbohydrates which are there and its functions. Okay, so this is the initial part which will give you 3 marks. You explain what a fluid mosaic model is, give the diagram of it, what are the various parts of it, the protein, lipid, carbohydrate, describe what are they, and what are the functions of each of them separately. Okay, now as a whole, we can talk about your functions in general. So that you can talk about, selective permeability. Okay, barrier between your ICF and ECF. Protection. Okay. So we have selective permeability, the barrier function, protection functions. Okay, so these would be the most important things you would talk about. Apart from that, various small components like shock absorption. Okay, and you can even talk about signal transduction, cell-to-cell communication. Okay, so like that, we can talk about various general functions. Okay, so this way you're getting your full 5 marks. This will go around 1.5 pages of an answer would be there in this. Okay, so that is that question. The next question, explain the different types of intercellular connections and their significance. So intercellular connections, you have to majorly talk about your gap junction. tight junction, adhering junction, desmosome, then hemidesmosome. Actually, the question was specifically intercellular connection. So the remaining two things like hemidesmosomes and focal adhesions, you don't have to talk about it. Okay, so I'd want you to talk about your gap junction, tight junction, adherent junction, desmosome. Draw the diagram of it, explain what is the unit of it. Okay, like gap junctions, for example, your connexons. Tight junctions, we have your Claudine or Claudine junctional adhesion molecules. Okay, in adhering junctions, desmosomes, we have something called cadherin, along with filaments like actin and intermediate filament. Okay, along with that, I want you to draw the diagram of it. Now, what are the functions of each of them separately? So, gap junction, ionic transfer. Also, it is a type of electrical synapse also. Okay, so transfer of ions, communication between cells. That is one function of your gap junction. Now, tight junction, it is for controlling your paracellular flow. The tightness of it makes it either leaky or a tight sort of a junction based on the tightness of that unit. So the paracellular flow is important over here. Adhering junction, what it does is it adheres the epithelial monolayer to make sure that it does not break off from there. Okay, so chances of metastasis and all that will be reduced because of adhering junctions, which make sure that the monomolecular layer is staying treated. Desmosomes are what prevents slowing of when a shearing stress is given. So when a shearing stress is given, epithelial, which are next to each other, should not just get ripped off. So response to a shearing stress is through desmosomes. Okay, so like that, I want you to describe what is the structure, what is the unit it's made of, draw a diagram for each of this, and talk about the function. So that will be more than enough to fetch you 5 marks for this. Okay. Next, discuss the process of apoptosis and it's important in maintaining health. So what is the process of apoptosis and importance in maintaining health? Here also I'll give it something like 3 and 2 marks. So first, process of apoptosis. So in process of apoptosis, you'll have to talk about your extrinsic and your intrinsic. And you have to talk about how it activates through death receptor caspase 8. And here, intrinsic either from mitochondrial damage or nuclear damage will eventually activate caspase 9. Okay, once these two are activated, finally it'll go to your executioner caspase. That is 3, 7. And this will finally form pores, which will result in death of the cell. Okay, so this is a programmed type of cell death where there is no inflammation, destruction of the entire area. It is localized and that tissue dies off, eventually becoming smaller apoptotic bodies. Okay, what is the importance of this? So that is the next part. Importance in maintaining cellular health. So, for example, if there is a intracellular parasite or virus, okay, or some prion is there, misfolded proteins are there. So these sort of things to kill off the cell individually without affecting everything else in that vicinity, we can go for apoptosis. Okay, apart from that, here you can also mention few other functions of apoptosis, like in your cellular health, for example, in menstrual cycle. It is through apoptosis that degradation of these various structures happen. That is another thing. Then your immune tolerance. Okay, so any T cells which are reactive towards self antigens needs to be selected and removed. Such clonal selection also is through apoptosis. And apart from that, again, during embryogenesis, how your digits are formed, in sexual differentiation. Okay, if you are a male, the remnant parts of the female tract should be regressed. That is also through apoptosis. Otherwise, what will happen if they keep remaining? Eventually, at some point in your life, they become teratogenic. Okay, so that is why these, again, these are all also answers for this question. Importance of maintaining cellular health. So if these structures remain, they are going to be causing harm in the body in the future. Okay, so that is why it's important for this to happen. So like that, if you mention all of these points, you're going to get 5 marks for this question. Next, describe the resting membrane potential and the factors that contribute to its establishment. So here, what you have to talk about first, you talk about what is resting membrane potential. So how you're going to answer it is, what is resting membrane potential? Okay, give its normal values. So I was explaining earlier, minus 17 for neurons, minus 19 large neurons, minus 19 skeletal and cardiac muscle, and in smooth muscles, how it is around minus 55. Okay, it's undulating type. So all of that you talk about RMP. Then you'll have to explain how RMP is generated. Here you have to talk about your potassium channels, which are leaky potassium channels. Then what is maintaining it? That is through your active sodium, potassium, ATPase. Okay, with diagram, explain this. Okay, because of lack of time, I don't think I can explain everything. Okay, so this is just a way to help guide you as to exactly what you're supposed to write. Okay, there's not enough time for me to go and explain everything. Okay, if there are any doubts, please be free to ask me. I can explain any specific thing if anybody needs any help. So again, you have to draw a diagram and explain all of this. So describe resting membrane potential and factors that contribute to its establishment. So there you talk about leaky potassium channels, you can talk about sodium potassium ATPase. Okay, then you over there are values which are given in Ganong, for example, where they talk about how much contribution by Goldman-Hodgkin's equation. Okay, that those values also you can write it down. You'll get better marks if you do all that. After that, the next question is explain the process of facilitated diffusion and how it differs from simple diffusion. So this is again pretty basic question. So you here again, you talk about what is facilitated diffusion. Like you can draw a tabular column on either side, each heading. Talk about what is facilitated diffusion. On the other side, what is simple diffusion. How you require a carrier protein in facilitated diffusion, how you do not require one. Both are down the concentration gradient. Both do not require ATP. And saturation kinetics is present in facilitated diffusion, which is not present in simple diffusion. You can draw a graph plotting your saturation kinetics. Okay, that also can be done. And with the diagram also, you can just, since it is a 5-mark question, otherwise, just this much should be like 3-mark question and all. Usually, this much is enough. Since it is a 5-mark question now, I'll want you to draw actually diagrams for each of this with examples. Okay, some example of facility division, some example of simple division. Then you'll get 5 marks. Okay, otherwise, with just that much, would only fetch you maximum 3 marks. So in order to get a 5-mark, you have to add these extra points to it, the diagram and the examples. Next question, describe the different body fluid compartment and their composition. So here you have to talk about the various body fluid compartments. So again, diagrammatically, you can explain everything. You can talk about your ICF. You can talk about your ECF, which is made up of your plasma and your interstitial fluid. Okay, then you talk about the various percentage of, for example, water, which is there. Okay, how, around 60% of water is there. Okay, 40% in your ICF, your whole ECF is 20. Okay, now out of that, just a minute. Okay, so 20 is over here. And out of that 20%, you have 5% and 15%. So that division you have to talk about. Okay, you can talk about 42 liter, this becoming your 20 liter, 28 liter, and this being some 14 liter. And here you can talk about 11 liter and 3 liter. Okay, so this basic thing you can talk about in terms of composition. Okay, then again, the ionic composition also you can talk about. So the ECF versus the ICF. The ionic composition, earlier I was telling you. So in the ICF, potassium, phosphate, protein. Okay, so these are the major cation and anion. In the ICF, here, sodium, chloride. Okay, there's a diagram in Canon, which is pretty good. You can use that actually. Okay, so describe the different body fluid compartment and their composition. And again, you can further talk about ICF. Again, since it is a 5-mark question. Okay, in the interstitial fluid, we have the extracellular matrix, which is there. Okay, what are the components of the matrix? You can talk about how type 1 collagen is present, proteoglycans are present. Okay, and elastic fibers are present. That is what is there in between these. All right. So you talk about the water composition, the ion composition, extracellular matrix composition. Okay, and what is there in between also? In the diagram, you can mention the epithelium, you can mention the endothelium, basal lamina, all of these also can be mentioned. Okay, so like that, you can discuss. Next is discuss the role of lymphocytes in the immune response. So for this answer, if you're talking about lymphocytes, we have, so since they're specifically asked only lymphocytes, we have your T cells and your B cells. Your T cells, you have your helper T cells. Helper T cells, we have type 1 and you have type 2. Apart from helper, you have the cytotoxic. So that is basically your CD4 plus and CD8 plus. On top of that, you can also mention your memory T cells and suppressor T cells. Okay. In B cells, you can talk about your how clones are formed. Okay. And with this, you try to explain how lymphocytes function. So, for example, you talk about a antigen being present. Okay. So that antigen is detected by an antigen presenting cell. Then your lymphocyte by itself can help in being an antigen presenting cell. Now, antigen presenting cell, from there, what will happen? Your direct activation of your cytotoxic T cells can happen. Okay. Or through helper T cells, either it can activate your cytotoxics again, or it can activate your B cells also indirectly. Okay. And what does memory cell and suppressor cell do? Memory cells helps form the clone for selection later. Okay. Your suppressor, that is what forms an area around this immune response to make sure that a widespread is not happening. Okay, so these are all different ways by which your lymphocytes are gonna help. Okay, please don't, write all the steps of addition, all of those steps, margination. That is generally for your neutrophils. And here the question is, okay, that is, I'm explaining this because I've seen students make these mistakes. So here the specific question is lymphocytes and immune response. How do lymphocytes act? Whatever I told you so far, that is how lymphocytes actually work. Okay. Next, explain the transport mechanisms involved in the movement of substances across the membrane. This is a very huge answer. Okay, but since it is a 5-mark question, you have to answer according to that. So here, again, you have to have a proper framework. You can talk about your transport across the cell membrane. So here, you'll have to talk about your simple diffusion. Okay, passive, like simple diffusion, facilitated diffusion, non-ionic. Again, since the 5-mark question only, don't describe them too much in detail. Okay. Then you talk about active, give examples for each of them. Primary and your secondary with examples. Okay. On top of that, go to vesicular transport, where you talk about your endocytosis, exocytosis. Okay, this is where you talk about your clathrin, caveolin, and you can talk about your phagocytosis, cytosis, pinocytosis. Okay, so all of these things needs to be described here. And since it is too much, just describe briefly. Okay, this whole thing should not go more than 2 pages for 5-mark question. Okay, just one diagram, example, that will be more than sufficient. And what it does, that will be more than sufficient. Okay, again, for a 5-mark question, don't like keep writing, especially since this would be something normally a 10-mark question. Right? Next, we'll go to your 3-mark questions. So first question is, what is the primary component of ECF? So some repetitions are there in these questions. Okay, so what are the primary components of extracellular fluid? So this is something that we answered earlier also. So in extracellular fluid, you have to talk about your plasma and the interstitial fluid and what are the components of it. Okay, in your plasma, what are the components of plasma? Since this is specifically asked, you can talk about how in the plasma, 91% is water. Okay, the remaining, we have organic and inorganic components. Out of that, plasma has plasma proteins in them and ions. Okay, and then you can just mention few plasma proteins and all. And then you talk about the interstitial fluid, and then the ionic composition difference between them and how proteins are lesser in the interstitial fluid. Okay, so all of these things you can draw and explain, and in even the interstitial fluid, you can talk about the extracellular fluid matrix also. Okay, so that will be more than sufficient for 3 marks. Next, define osmolarity and its significance in body fluid. So again, please be wary, difference between osmolarity and osmolality. Okay, so the concentration of the number of particles which are there in liter of the solution, that is osmolarity. Osmolality will be per kg of solvent. Okay, so that basic difference is there. So that is the definition. And then significance in body fluid regulation. So osmolarity, what does it do in body fluid regulation? Osmolarity, based on that, for example, something as basic as behavioral modification. Thirst. Okay, so that is one of the first things that is going to happen. Osmolarity increases your behavioral modification, increase thirst, and you drinking water. On top of that, there will be release of ADH. Okay, so that is also going to be there. Okay, then the fluid balance can be adjusted based on that. Okay, so this is different ways by which your osmolarity is going to affect your body fluid regulation. Okay, and also how it affects the potassium movement in your nephron, in the distal tubules. That is also another thing. So these are various ways by which osmolarity affects your body fluid regulation. And actually you can, talk about the osmoreceptors, where they are, and draw a diagram and explain all of this. Again, not much is needed because it's a 3-mark question. 1 mark would be definition and 2 marks to describe the significance of body fluid regulation. Next is explain the role of sodium potassium ATPase in maintaining membrane protection. This is very specific. Okay, sodium potassium ATPase is a very big question to deal with. But here they're specifically asking how does it maintain membrane potential. So earlier we were talking about it. Okay, so that explanation you'll have to give now. How, leaky potassium channels generate your RMP. Okay, and sometimes in between, say for example, some leaky positively charged ions through non-selective channels or any other way, calcium, anything comes inside. Okay, now what happens is, because of this is moving towards the threshold. But as long as this is not reaching threshold, it is a graded potential, it'll come back to your RMP. How does that happen? So that is where your sodium potassium ATPase is there, which takes this sodium outside against concentration gradient and brings potassium inside. And it's not same because this is 3 and this is 2. So net effect is a less positively charged ions inside. So this is one way by which your sodium potassium ATPase helps maintain your membrane potential. All right. Now, what are tight junctions and the functions in epithelial tissue? So here again, 3 marks separately, since it is asked, you'll have to draw the diagram properly and explain. And function again, paracellular flow. We talked about it. Okay, we talked about the paracellular flow. Just give me a minute. Okay, my charge is running out. So again, we'll have to talk about the structure. Okay, talk about your zona occludens structure. Okay, junction adhesion molecules and how it is finally linked with your actin and how it is there on either side. Draw the diagram. Okay, so what is the tight junction? Draw the diagram, explain what it does. Again, in the diagram only you can explain it. Okay, that will fetch you 3 marks. Explain the concept of active transport with an example. Again, you can talk about sodium, potassium ATPs. Okay, so for active transport, sodium, potassium ATPs you can use, or your proton pump for gastric acid secretion, anything you can use. Okay, even circa for that matter. Okay, so all of these are examples, so you can use any of this, even your sodium, potassium to chloride channel. So you can use anything. Okay, active transport, whatever is there, you can use it. Okay, and they have not even specified whether it should be primary or secondary. Okay, so first talk about movement against concentration gradient, how ATP is required. Okay, that is what constitutes active. And under active, mention how primary, secondary is there. Okay, and then give an example and explain. All right. Next is describe the role of hemoglobin in oxygen transport. So here you can talk about hemoglobin. You can discuss your Bohr effect, Haldane effect. Okay. Since it's just a 3-mark question, that would suffice. Bohr, Haldane, and maybe you can talk about your hemoglobin oxygen dissociation curve. Okay. Lightly. Okay. So that will be more than sufficient for 3 marks. If it comes for a higher mark, then you can talk about your cooperative binding, salt bridges, okay, how they break to 3-DPG. Okay, so all of that you include it if it is a 5-mark question. Otherwise, if it's a 3-mark question, I think role of hemoglobin, you can just explain it with and specifically oxygen also, not even carbon dioxide, just oxygen. Okay, so it becomes very simple. You just draw 2 diagrams, one describing your Bohr effect, the other describing your Haldane effect, and that'll be more than sufficient for this. Okay. What is phagocytosis and its role in immunity? So what is phagocytosis? First thing. Second point, what are the steps in phagocytosis? So you have to talk about the size of this substance that needs to be phagocytosed. Okay, so it should be more than 0.5 micrometer. Okay, then the steps, how detection, then how engulfing is happening, how phagosome is being formed, and then phagolysosome is formed. Okay, and the ultimate destruction and exocytosis. So these are various steps that are involved. With diagrams, you explain all this. And role in immunity, 1 mark. So major thing will be what is phagocytosis, explaining it, and how does it help in immunity. So substances, foreign objects, not only foreign objects, even things inside, which needs to be degraded, which is more than 0.5 micrometer, is through phagocytosis. Okay. Define the term cell signaling. So what is cell signaling? The way a body cell responds. Okay, so some stimulus or any changes there or chemical mediator, whatever it is, or cell-to-cell signaling, no matter what it is, how a cell responds by using certain signals which are present either on the surface of the membrane or inside the membrane. That is what we call cell signaling. Okay, your examples, you can use a ligand-gated ion channel. Simple, that is a simple example, ligand-gated ion channels. Or you can talk about your enzyme linked activity, G protein coupled receptors. Okay, these are all different examples that you can provide with description. Explain the significance of calcium ions in cellular function. So calcium in cellular functions, you can talk about your muscle activity. Okay, so with a diagram, describe how calcium comes in, how it binds specifically separately in smooth muscle and in your cardiac muscle and your skeletal muscle. It's different, extracellular, whether it is intracellular. Okay, and how the whole mechanism instead of troponin, tropomyosin, that instead of that in your smooth muscles, it is calcium calmodulin pathway, myosin light chain kinase. So all of that with diagrams, three things you describe one thing. Okay, then you talk about importance of calcium in your hemostasis, in clot formation, in which stages calcium is required. Okay, then you talk about how it is helpful in your teeth, your bone mineralization. Okay, then also the importance of calcium by itself as a signaling mechanism. Okay, so calcium influx by itself can be a signaling mechanism in various. So with example, you provide that. Then you talk about how it is important in synapse and neuromuscular junctions also. Okay, calcium is involved over there. Okay, and you know, you can just write one. You don't have to describe clinical. You can write a heading clinical, and you can talk about how Lambert-Eaton syndrome, when this gets affected in the presynaptic terminal. Just an example of a clinical scenario that will gonna give you 3 marks for this. Okay. Next is what is an antigen? So antigen is usually a substance which evokes immunogenicity. Okay, so what happens is the substance is detected by your immune cells, and in response to that, immunogenicity is there. Okay, so and how does this happen? Earlier, again, the same thing I was explaining to you. Your helper T cells, antigen presenting cells, then helper T cells. Okay, and either through humoral pathway or cytotoxic pathway, how it mediates. So it's actually a repetition that is coming. Okay, so that is how you describe that. Now, coming to your 10-mark question. Explain the principle of homeostasis. So here you have to talk about what is homeostasis. Okay, you can talk about Claude Bernard, milieu interior, all these concepts you can talk about. Okay, what is homeostasis? You can mention the receptor, the central organ, as well as the effector and how it changes. Also, I want you to talk about gain, concept called gain also in this. Okay, so gain is correction by error. So that is also something, because it's a 10-mark question. Okay, then discuss the mechanisms by which the body maintains homeostasis. How can it do that? Either to a negative feedback mechanism or a positive feedback mechanism. Usually, homeostasis is maintained by either negative or a positive feedback mechanism. So, describe what is the negative feedback mechanism, describe what is the positive feedback mechanism. Okay, with example. So, for each mechanism, give at least 2 examples with flowcharts. Okay, because it is a 10-mark question. All right, so this should fill around 2.5 to 3 pages. Okay, so when I say page, I'm talking about, each side separately. Then we have the next question. Describe the composition and functions of blood. Discuss the role of different components of blood in immunity. So this again is a big question. Okay, and 10 marks, but there is a lot to write under this. So what do you do in this? Describe the composition. Okay, when you talk about blood, you talk about, okay, again, you talk about you can talk about the PCV, how there is plasma, you have cellular elements. Okay, what are the 3 different parts of the cellular elements? Okay, so that you can draw and explain. Okay, and apart from that, I was talking about the 91% water, the organic component, inorganic component, 7%, 1%, and how plasma proteins are there. Talk about what are the different types of plasma proteins. Okay, so plasma proteins, RBC, your WBCs, and your platelets, cellular components, along with the plasma and the fluid, all of these. Okay, so these are all the different composition. Then a general heading of functions of blood. So basically, when you talk about functions of blood, in that you can talk about functions of your plasma proteins, functions of your hemoglobin. Okay, so all of these are separate, which can be put together to give the functions of blood. So functions of blood, transport of oxygen, carbon dioxide. Okay, transport of all of this. Hemoglobin function, acid-base balance. Okay, then you can talk about the functions of plasma proteins specifically, major ones. You can talk about your colored osmotic pressure. Okay, you can talk about your hemostasis, fibrinogen, globulins, immunoglobulins with its diagram. Explain that. Okay, then talk about how it is a reservoir of proteins, how it is important in transport. Okay, so at least these five things are very important. On top of that, you can talk about acid-base balance, ESR, maintenance of ESR, viscosity. Okay, so all of these functions of plasma proteins along with functions of your hemoglobin. Okay. On top of that, discuss the role of different components of blood in immunity. So here again, immunoglobulins, plasma proteins, immunoglobulins, how it helps in immunity. Draw the diagram and explain that. On top of that, we have your WBC, which are lymphocytes, functions of lymphocytes. Eosinophil, draw the diagram, functions of eosinophil. Basophils, again draw the diagram, functions of basophil. Monocytes, draw the diagram, functions of monocyte. Neutrophils, draw the diagram, again, functions of it. So you see how big a question this is. Okay, so you have to, accordingly not go too deep into everything. So just as I was telling, diagram, function. Okay, that'll be enough for discuss the role of different components of blood in immunity. On top of that, one, point wise, you can talk about the functions of blood and composition. Okay, so this is just a layout as to how you have to approach a question if it is coming like this, all of these questions. So the major problem would be, again, trying to finish it time. Okay, so again, these are the various features of those of you who are not aware of this. These are various core features of Oracle. We've got live sessions which are available. Pre-recorded sessions are there which can be used and downloaded anytime. We have a comprehensive list of notes for various subjects which are there, as well as MCQs, which will help you prepare for your exams. These are the various subscription plans which are currently available. And you can use my code Dr. Shakti20 for discount and subscription. Okay, and these are the various social media handles if you want to communicate with the team in Oracle for any reason. Okay, and notes and all are usually given out in the Telegram app. Okay, so if any doubt is there, please let me know. Any difficulty regarding how you have to answer a question, any topic which I need to explain in particular, please let me know. Okay, so I guess there's nothing. So I'll see you again next session.